Good morning everyone. Welcome to your biology class. This video is in continuity with the previous video of the chapter cellular respiration, and in this video we will talk about the Krebs cycle. So the main objectives we are covering in this video are steps involved in Krebs cycle and significance of Krebs cycle. So first of all, Krebs cycle or citric acid cycle. So the name Krebs cycle is given after the name of the discoverer Sir Hans Adolf Krebs. Krebs cycle is also known as the citric acid cycle because it begins with the formation of citric acid, or as the tricarboxylic acid cycle because citric acid has 3 carboxyl groups. Krebs cycle takes place in mitochondria. Next, this cycle only occurs under aerobic conditions as energy-rich molecules like NAD positive plus FAD can only be retrieved from their reduced form once they transfer electrons to molecular oxygen. The citric acid cycle is the final common pathway for the oxidation of all biomolecules, proteins, fatty acids, carbohydrates. Molecules from other cycles and pathways enter this cycle through acetyl coenzyme A. The citric acid cycle is a cyclic sequence of reactions formed of 8 enzyme-mediated reactions. This cycle is also particularly important as it provides electrons, high-energy molecules to the electron transport chain for the production of ATPs and water. Pyruvate formed at the end of glycolysis is the first oxidized into acetyl coenzyme A and then enters the citric acid cycle. So here the steps of Krebs cycle. So first of all, this is the overall equation or the reaction of the citric acid cycle. Then in eukaryotic cells, the enzymes that catalyze the reactions of the citric acid cycle are present in the matrix of the mitochondria, except for succinate dehydrogenase and aconitase, which are present in the inner mitochondrial membrane. One common characteristic in all the enzymes involved in the citric acid cycle is that nearly all of them require magnesium ions. Now, oxidation of pyruvic acid to acetyl coenzyme A. So after glycolysis in aerobic organisms, the pyruvate molecules are carboxylated to form acetyl coenzyme A and carbon dioxide. Oxidative decarboxylation of pyruvate to acetyl coenzyme A. It is an irreversible oxidative decarboxylation reaction in which a molecule of carbon in the form of carbon dioxide is removed from pyruvate. In this reaction, a molecule of pyruvate generates one NADH molecule. This step is linked between glycolysis and Krebs cycle. Then first step, condensation of acetyl coenzyme A with oxaloacetate. So it is a condensation reaction. Acetyl coenzyme A condensed with oxaloacetate to form citrate, and the reaction is catalyzed by the enzyme citrate synthase. Oxaloacetate play catalytic role in citric acid cycle, and at the end of process, oxaloacetate is regenerated. Second step of Krebs cycle is isomerization of citrate into isocitrate. So the enzyme aconitase catalyzes the isomerization of citrate to isocitrate with intermediate cis-aconitate. This is a reversible reaction. This reaction occurs in 2 steps. First is dehydration and second hydration. Then third step of Krebs cycle involves oxidative decarboxylations of isocitrate. So the enzyme isocitrate dehydrogenase catalyzes the oxidation of isocitrate to form alpha-ketoglutarate and carbon dioxide. In this, step 1 molecule of NADH is generated. It is an irreversible reaction. Then step 4, which involves oxidative decarboxylation of alpha-ketoglutarate. So this is an oxidative decarboxylation reaction catalyzed by alpha-ketoglutarate dehydrogenase enzyme in which alpha-ketoglutarate is oxidized into succinyl coenzyme A and carbon dioxide. In this reaction, coenzyme A serves as carriers of succinyl group and NAD serves as electron acceptor. One molecule of NADH is generated in this step. Now, step 5, which involves conversion of succinyl coenzyme A into succinate. So, conversion of succinyl coenzyme A to succinate is catalyzed by succinyl coenzyme A synthase or succinic thiokinase enzyme. This is a substrate level phosphorylation reaction in which coenzyme A group ultimately donates its phosphate group to GDP, forming energy-rich GTP. A molecule of carbon dioxide is released in this step. Step 6, that is dehydration of succinate to fumarate. So, succinate dehydrogenase catalyzed the oxidation of succinate to form fumarate. It is a reversible reaction. In this step, a molecule of FADH2 is generated. Then step 7, that is hydration of fumarate to malate. So, this reaction is catalyzed by fumarase, that is fumarate hydratase, in which fumarate is converted into malate. This is a hydration and reversible reaction. Step 8, dehydrogenation of L-malate to oxaloacetate. Malate is oxidized into oxaloacetate, generating a molecule of NADH. This reaction is catalyzed by malate dehydrogenase enzyme. Oxaloacetate is regenerated in this step and combines with acetyl coenzyme A and continues the cycle. So here, this is the Krebs cycle in detail. Now next is significance of Krebs cycle. Now the role of TCA cycle, first of all, role in central metabolic pathway. So tricarboxylic acid cycle is a final common metabolic pathway of carbohydrates, fatty acids and amino acids. At first, all these biomolecules are catabolized by their separate metabolic pathways to generate acetyl coenzyme A, and then acetyl coenzyme A enters TCA cycle for further metabolism in aerobic condition. TCA is more efficient in energy conservation than other pathways of metabolism. Second, TCA is amphibolic pathway. So it plays a role in both catabolism and anabolism. Catabolic role. So TCA is a catabolic pathway because it oxidizes acetyl coenzyme A completely into carbon dioxide and water and releases large amount of energy. And it is an anabolic pathway. So TCA is an anabolic pathway because it provides precursors for biosynthesis of other molecules in cells, such as citrate, alpha-ketoglutarate, succinyl coenzyme A and oxaloacetate, which acts as precursors for biosynthesis of various molecules. Glucose, purine and pyrimidine are synthesized from oxaloacetate. Fatty acids and steroids are synthesized from succinyl coenzyme A, and some amino acids, purine and pyrimidine, are synthesized from alpha-ketoglutarate. So citric acid cycle is an aerobic process. So NADH and FAD are electron acceptors in the TCA cycle. These are regenerated by electron transport chain which requires oxygen as final electron acceptor. Hence, overall TCA and electron transport chain are aerobic processes. Thank you.